Data contamination is when evaluation material, or close echoes of it, leaks into a model's training or tuning pipeline, so the model looks smart by remembering the test instead of generalizing the skill. It happens in several quiet ways. Benchmarks and answer keys get scraped into pre-training corpora. Near duplicates appear as paraphrases, translations, or template variants. Dataset formats and prompt patterns teach the quiz rather than the competency. Supervised tuning and RLHF recycle good examples that contain benchmark items, and post-deployment log ingestion drags test content back into retraining. The result is inflated scores, misleading model comparisons, and brittle behavior that collapses off distribution. Detecting it is hard because corpora are opaque and evolving, paraphrase and format cues evade string match checks, and exposure can be split-specific or occur after release. In short, contamination turns evaluations into mirages. In this lecture, I will introduce SCAR, Spectral Contamination and Alignment Rupture, a geometry-first way to tell whether a model is thinking or replaying. The problem is simple to state and hard to see. Benchmarks leak, shortcuts creep in, and glossy answers hide fragile reasoning. SCAR stops grading the final sentence and starts reading the trajectory that produced it. We watch how the internal spectrum bends, how much effort the network expends as it moves through layers, and how its belief direction is steered turn by turn. When performance is honest, these paths are smooth and purposeful. When contamination or shortcutting is at play, the paths fracture, late layer spikes in spectral curvature, sudden dips in path effort, and oversteered belief vectors that snap toward memorized anchors. That fracture is the rupture in SCAR. Practically, SCAR gives you a set of inspection tools you can read at a glance. A karyotype band map shows where curvature, effort, and drift coactivate in the same layer. A 3D isosurface caldera reveals thick rims of concentrated evidence in contaminated models versus thin, broken shells when clean. A semantic earthquake trace marks uplift points along depth, epicenters of instability. A volcanic landscape surfaces the final per-layer eruptions after multiple signals agree. These views are paraphrase robust, split-aware, and come with simple reporting so results can be reproduced and audited. Why this matters: inflated scores waste research time, mislead customers, and push safety decisions onto sand. SCAR restores signal. It separates generalization from memorization and stable alignment from cosmetic coherence, using measurements you can automate and thresholds you can defend. By the end of this talk, you'll know how to spot rupture layers, compare checkpoints, and plug SCAR into your eval pipeline so great numbers actually mean great models. In recent time, data contamination has moved from a niche worry to a central threat to credible NLP evaluation. Benchmarks that once served as gold standards are turning up, verbatim, paraphrased, or template, inside training and tuning corpora, inflating scores and distorting model comparisons. The position paper, NLP Evaluation in Trouble, on the need to measure LLM data contamination for each benchmark crystallized this problem. It argues that contamination is widespread yet undermeasured, that its impact is uneven across splits and model stages, pre-training, fine-tuning, alignment, even post-deployment, and that future papers should not claim state-of-the-art without split-level contamination checks. The authors propose a community protocol, clear labels, contaminated, suspicious, clean, reproducible evidence, and public registries like the LM Contamination Index to replace guesswork with auditable facts. Our work builds on that call. We adopt the same accountability, but go one step deeper with SCAR, reading the model's internal geometry to detect contamination even when string matches fail. The LM Contamination Index is a curated registry that documents where evaluation datasets have leaked into language model training, tuning, or retraining pipelines. For each model corpus dataset split, it labels status as contaminated, suspicious, clean, or not applicable, and cites the evidence source, paper, or community report. Use it as a reliability lens. Contaminated or suspicious splits likely inflate scores by memorization, while clean splits better reflect true generalization. Let us have a quick look on LM Contamination Index. Natural language evaluation is facing a quiet crisis. Many public benchmarks have slipped into training data, so some model scores reflect memory, not true skill. The LM Contamination Index is a simple guide to that risk. It lists common corpora and models, and labels each dataset split as contaminated, suspicious, clean, or not available. Contaminated means we found hard evidence. Either the dataset appears in the pre-training corpus with a measured overlap, or a model trained on that corpus can regenerate examples. Suspicious means there are warning signs. The model seems to know the dataset partially, but we do not have a direct match. Clean means no signs or evidence were found. Not available means the split is private. A blank cell means no test was run. Use the source column to see where the claim came from. Repository marks are community reports. Treat high scores on contaminated or suspicious splits with caution. Favor clean splits when you compare models. That is how we keep evaluation honest and progress real. Why do we need SCAR? Because contamination detection is hard in all the ways that matter. First, the data are opaque. Closed models rarely disclose corpora, and even open models ship evolving snapshots, so clean versus contaminated is guesswork without provenance. Second, leakage isn't just copy-paste. Benchmarks sneak in as paraphrases, translations, template prompts, or synthetic variants that carry the same answer key well beyond what string matching can catch. Third, exposure is granular. A single split or a thin slice of a benchmark can leak while the rest remains clean, so dataset-wide labels blur the truth. Fourth, models don't freeze after launch. RLHF refreshes, feedback replay, and data updates can retroactively contaminate an otherwise clean release. And finally, at scale, audits are expensive and brittle, versions fork, mirrors drift, thresholds vary, and reporting is inconsistent across papers. SCAR answers with standardization and structure. It gives us a common taxonomy and thresholds, clear, split-level labels for contaminated, suspicious, and clean, with minimum evidence rules. It defines a unified protocol for both open and closed models, corpus scans, paraphrase and translation checks, memorization probes, and post-deployment screens. It ships a robust probe suite that goes beyond string match, prefix extractability, templatic prompts, and cross-lingual audits to surface non-verbatim leakage. It reports split-aware metrics with confidence intervals, so we can point to exactly where exposure occurs. It maintains a version registry, signed results with dataset IDs, hashes, timestamps, and model versions, so findings are traceable and comparable. And it integrates with real workflows via automation and governance, evidence cards for reproducibility, and simple policies so papers and releases carry SCAR scorecards by default. In short, SCAR turns contamination detection from ad hoc forensics into a repeatable, auditable, decision-grade process. Now, let's see SCAR in action through 5 complementary lenses, each measuring rupture from a different angle. We start with nDNA trajectories to watch the belief path turn by turn. We plot a path through the network, layer by layer. For each layer, we track 3 things. Curvature, how sharply the path turns. Thermodynamic length, how much effort the model spends to move forward. Step size, how big the update is between layers. Clean models draw canalized paths, gentle turns, steady effort, and regular step sizes. Contaminated models show kinks, cusps, or tight spirals. The path bends suddenly while effort drops. That pattern means the model is jumping to a memorized anchor instead of reasoning through the layers. To make this easy to read, we also show 3 simple projections. Curvature versus effort. Healthy behavior lives in the moderate turn. Steady effort zone. Big turns with low effort signal a shortcut. Curvature versus step size. Big turns with tiny steps suggest oversteering toward a memory. Effort versus depth. A trough in the late layers marks the likely rupture zone. Bottom line for narration. Smooth arc means generalization. Abrupt bend plus effort dip means rupture. We now use 4 more lenses to pin it down. Karyotype bands coactivate. For each layer, we show 3 bars, curvature, effort, and drift. If all 3 rise together in the same late layer, that's a coactivation event. Isosurface caldera. We render a 3-dimensional shell of high evidence. Clean runs show thin, broken shells. Contaminated runs show a thick rim that hugs the late layers. A slow spin of the figure is enough to tell shallow from volcanic. Semantic earthquake map. This is a depth-wise uplift trace with confidence shading. Positive peaks mark epicenters that match the karyotype bands. Read it like a seismogram. Small ripples are noise, a sharp, significant crest fixes the rupture layer. SCAR volcanic landscape. We collapse all signals into a calibrated score per layer. Local peaks that pass the statistical filter are called eruptions. Gentle shelves suggest clean behavior. Sharp spikes in late layers confirm contamination. This gives a quick verdict for reports and dashboards. Bottom line. Watch the path. Confirm coactivation. See the 3D shape. Pinpoint the depth. Issue the per layer verdict. 5 views tells one unified story. Smooth arcs is equal to generalization. Late layer eruptions is equal to rupture. Let's us now see all of them in action one by one. On the right you see our karyotype, a 3D bar panel by layer. Each layer gets 3 bars, red for curvature spike, blue for effort dip, and green for drift surge. Bar height is normalized so we can compare models. Small black cubes mark layers that pass our statistical test after FDR, that is false discovery rate correction. How do we read it? A clean karyotype shows balanced, mid-height bars spread across mid and late layers. There is no single layer where all colors jump together. A contaminated karyotype shows coactivation, a tall red band that sits next to a tall blue band and a tall green band in the same late layer. That trio is a rupture band. It matches what we saw in the trajectory view: abrupt bends, depressed effort, and a strong steering pull. Diagnostics: the width and height of the bands track rupture strength. If only one color rises, that suggests a confound, for example, a quantization change that alters effort without real curvature. When all 3 rise together, the signal is structural, not cosmetic. Takeaway: this karyotype is a fast, layer-by-layer scan. One tall trio is a warning. A stack of adjacent trios forms a rupture corridor and calls for deeper audit. Think of this like an MRI for the model. In medicine, you scan a 3-dimensional volume, draw the boundary around the brightest region, and trace the crest to find what matters. We do the same here. Our volume is an evidence field built from the model. One axis is layer depth, one is a representation slice, and one separates clean versus suspect runs. From that volume, we draw a semi-transparent shell around the strongest evidence. Thicker and denser parts mean stronger signal. On that shell, we trace a ridgeline, the high ground where the signal peaks. That ridge often looks like the rim of a crater. How to read it? Clean model. The shell is thin and patchy. The ridge is broken. There is no closed ring. Contaminated model. The shell is thick and continuous near the late layers. The ridge forms a closed ring. We mark a rupture layer on that ring so you can report the exact layer index. Why this works? Same logic as MRI: volume, then boundary, then ridge, then diagnosis. One slow spin answers the key question: is the structure shallow and fragmented, or deep, crater-like, and anchored near the end of the network? This view is robust to paraphrases and cross-checks with the other lenses. The closed ring aligns with karyotype coactivation and peaks in the semantic earthquake map. Bottom line: thin, broken shell means clean. Thick, closed rim in late layers means rupture. This view treats the network like a landscape and asks a simple question: where does the ground lift up? We compute a smooth baseline across layers and then measure how much the evidence surface rises or sinks relative to that baseline. The result is a ground displacement map. How to read it? Warm colors show uplift. That is a convex bulge in evidence, the signature of a rupture forming. Cool colors show subsidence. That is a dip, usually benign. The x-axis is the layer index. The y-axis lists models or runs. The vertical axis is the displacement value. Clean models show shallow undulations with no sustained bulge. Contaminated siblings show a sharp uplift concentrated in late layers. The uplift is stationary across paraphrases and format swaps, which tells us this is structure, not just wording. We mark significant epicenters with light markers, and we place a tick at the rupture layer so you can quote a single index. Volcanic landscape. This view combines signals into one surface so we can spot instability at a glance. For each layer, we aggregate curvature spike, effort dip, drift surge, and uplift into a calibrated score. We then look for local peaks. Peaks that survive the statistical filter are marked as eruptions. An eruption is a layer that consistently behaves like a hotspot. How to read it? The front axis lists the models or runs. The side axis is layer depth. The height is the combined score. Translucent bands show confidence around the ridge. A clean model shows a gentle shelf, no sharp peaks, no sudden cliffs. A contaminated model shows a tall, late layer spike. That spike is the eruption, and its label gives the rupture layer you can report. Focus on the late layers. The tallest spike you see here is the instability point. Notice how this spike lines up with the closed rim we saw in the isosurface caldera and with the warm uplift crest in the semantic earthquake map. When this surface stays smooth with only low bumps, we report no eruption, which supports a clean read. Bottom line, smooth shelves suggest robust reasoning. Tall, late layer eruptions indicate rupture and likely contamination. SCAR framework at a glance. Here's how SCAR runs end-to-end. Input. We start with a trained transformer and a set of prompts to probe the model, and we also use paraphrase variants. Inference settings are fixed, greedy decoding, temperature 0. We cache activations and lock a seed so the run is reproducible. Stage 1, load and extract. For every layer and every prompt we collect centered activations and gradients. From these we compute 3 per layer signals, spectral curvature, thermodynamic length as an effort measure, and belief drift magnitude as step size. Stage 2, calibrate. We standardize each signal with robust stats, median and MAD, so layers and families are comparable. We apply a light 3-layer smoothing and set split-aware thresholds from clean anchors. The goal is a calibrated scale that travels across models and prompts. Stage 3, detect with FDR control. We build a composite evidence field that rewards coactivation, curvature spike with effort dip and drift surge. We scan for contiguous bands of high evidence, compute per-layer p-values, and apply Benjamini-Hochberg to control the false discovery rate. The leftmost maximum inside an accepted band is the rupture layer. Stage 4, score and report. We produce scalar scores, SCAR as the max evidence and SCAR area as the integrated strength. We export layer-wise plots, nDNA trajectories, karyotype bars, the semantic earthquake map uplift, the isosurface caldera, and the volcanic surface. We also attach an audit card, thresholds, smoothing, seeds, permutations, and paraphrase stability. Interpretation. The arrows on this chart mean data flow from model to geometry, then to calibrated evidence, then to statistically significant rupture bands, and finally to a clean reportable score. The pipeline is paraphrase robust, split-aware, and reproducible. If the same late layer lights up across views and passes FDR, we call it a rupture. If not, we call it clean. What you see here. On top is a 3D line plot of SCAR score versus layer for 3 models. The horizontal plane lists the model family. The side axis is the layer index. The vertical axis is the calibrated SCAR score. Each point is a layer. The line shows how evidence builds as depth increases. How to read the curves. Llama, clean, stays low and steady, with only small bumps. That is healthy background noise. Alpaca, leaked, climbs late, forming a gentle ramp in the final layers. That is a suspicious pattern. GPTJ, contaminated, rises earlier and higher, then holds elevation in late layers. That is concerning and consistent with a rupture corridor. As a rule of thumb, flat and low means clean, a late climb merits review, and early, tall rise signals high risk. Now the heat map below. This matrix breaks the score into 3 causes across layers. Spurious recall, bottom row replay of seen items. Injection trigger, middle row prompt or template steering. Semantic drift, top row belief state pulled off course. Columns are layers. Dark cells mean weak evidence. Bright cells mean strong, calibrated evidence after robust normalization. Small markers indicate statistical significance with false discovery control. A dotted tick marks the rupture layer you can cite. What patterns to call out? In clean runs you see scattered, low-intensity cells with no vertical band. In leaked or contaminated runs you see a bright late layer stripe across rows. That stripe aligns with the ramp in the top plot. If the bright cells persist under paraphrase and format swaps, the signal is structural, not a wording trick. Top, how risk builds over depth. Bottom, what is causing it and where. Llama looks stable. Alpaca shows a late rise driven mostly by semantic drift with some trigger influence. GPTJ shows an early, strong rise with a clear late layer band and significant cells, fixing the rupture depth. Takeaway. Low, even trajectories and muted heat maps support clean. A concentrated late layer band with significant cells plus a rising trajectory supports a rupture call. So far, we argued for a shift in how we judge models. Don't stare at outputs and guess. Look inside the geometry where beliefs live. SCAR does exactly that. It finds the circuitry that carries contamination signatures, maps the surrounding field, and tests whether those signals hold up under paraphrase and split changes. The pipeline is simple to say and careful to run: measure curvature, measure effort, measure belief drift, then test for a real rupture while controlling false discoveries. The result is not a vibe. It is a layer index, a confidence, and a short story you can audit. From behavior to mechanism. The central idea is structural. Contamination is not only a string match, it is a shape in representation space. Across families and formats, SCAR keeps detection power without chasing wording. Because we localize the search, anchor it to clean exemplars, and spend an explicit drift budget, the method is stable under paraphrase, format shifts, and family changes. When SCAR says rupture, you can point to the layer, show the coactivation, spin the caldera, and replay the uplift. When it says clean, you can show why. What to carry forward? Locality with budgets. Search inside the causally implicated subspace and track how much drift you allow. This cuts false positives dramatically. Anchors beat absolutes. Calibrate against principled clean anchors, not a single global threshold. It travels better. Traceable causality. The audit trail, curvature to effort to drift to significance, makes failure modes legible. If detection fails, you learn whether it was mislocalized, misanchored, or overconfident. Compositional analysis. Treat contamination types as a sequence, not a one-shot label. Stable signatures emerge when you respect that structure. Final takeaway. SCAR turns a vague suspicion, maybe the benchmark leaked, into a mechanistic diagnosis you can defend, where it happens, how strong it is, and why we believe it. That is how we move from clever demos to trustworthy evaluation. The SCAR framework draws deep conceptual motivation from biological systems, where genomic integrity and regulatory balance are crucial for healthy function. Just as the overexpression of a single gene can lead to cancer or autoimmune disorders, contamination in LLMs causes the overactivation of memorized latent pathways, bypassing robust reasoning. The model does not understand, it simply remembers too well. This shortcutting behavior resembles oncogenic triggers. Minor local shifts in regulatory structure produce cascading effects on system-wide behavior. By treating layer-ways activations as a form of neural genomics, SCAR enables the detection of such epistemic mutations, offering a path toward more trustworthy and biologically inspired interpretability in AI. Let's start with a short primer, a quick video on gene overexpression and Waddington's epigenetic landscape. It shows how small pushes can tip a system out of its valley. Perfect intuition for what overactivation means in foundation models.
All living organisms construct proteins and enzymes using information encoded in DNA. To regulate the changing needs of the organism, genes along the DNA molecule are expressed or suppressed, depending on the cellular context. Epigenetics examines the complex inputs which chemically modify the structure of chromatin to render it more or less accessible in a heritable manner. For example, a methyl group can be added to or removed from histones, which may silence or activate a gene. Therefore, in the presence of polycomb, genes are silenced, whereas loss of polycomb opens up chromatin and induces transcription. The environmental characteristics navigated by the developing cells is demonstrated by the Waddington landscape, an illustration of how regulatory factors influence developmental paths. These dynamic and adaptive gene responses are a normal part of our physiology and development, but like mutations in the genes themselves, they can also go wrong. The prevailing model of cancer formation emphasizes somatic cell mutations, or mutations in cells which are not eggs or sperm. This somatic mutation theory proposes that a succession of several DNA mutations in a single cell causes cancer. A recent experiment on Drosophila fruit flies explored cancer generation via an entirely epigenetic mechanism. inspired by epigenetic research conducted over the last 2 decades. By temporarily reducing the activity of PRC1, an important epigenetic regulatory protein complex, researchers induced cancer in Drosophila cells. Even when PRC1 activity was restored after 48 hours, the altered cells retained their cancerous traits. This temporary PRC1 depletion triggered the induction of several genes, including signaling components that drive cell proliferation, dedifferentiation, and migration. These genes created a self-sustaining expression which continued even after the PRC1 complex was restored. Therefore, cells with a DNA compatible with normal development can experience a transient perturbation, leading to the formation of a cancer. These findings, along with the contributions of other research groups, warrant a reconsideration of the somatic mutation theory. The fact that initial cancer-causing events might be epigenetic could inspire new research and novel therapeutic strategies.
What the video teaches, biology side. Cells run on regulated pathways. Signals flow through receptors, kinasas, and transcription factors. Overexpression or a driver mutation bypasses control. A small tweak becomes a runaway cascade. Healthy tissue has checkpoints and feedback. Cancer forms when those controls are overridden and the system locks into a new state. The parallel in LLMs, transformers also have regulated pathways. Layers, heads, and MLPs route information along preferred subspaces. Contamination acts like overexpression. A benchmark pattern or answer key overactivates a memorized latent route. The model stops reasoning and just recalls. The result is a state switch. Belief dynamics snap from work through the problem to fetch the memorized template. SCAR's readout is basically the lab tests. In biology, you confirm disease with multimodal evidence, imaging, labs, and histology. One number is not enough. SCAR does the same inside the model. nDNA trajectories show the state switch, smooth arcs in clean models, kinks and cusps when a shortcut takes over. Karyotype bands catch coactivation of 3 signals in the same late layer, curvature spike, effort dip, and drift surge. That is your overexpressed pathway. The isosurface caldera is your MRI. A thin, broken shell looks benign. A thick, closed rim in late layers marks a lesion-like rupture ring. The semantic earthquake map is the uplift scan. Positive displacement is tissue bulging. Here it is semantic uplift at the rupture layer. The volcanic surface is like a volcano plot in genomics. A tall late layer peak is an instability hotspot. Why the cascade matters. In the video, one hot node, an oncogene, pulls the whole network into a new operating regime. In contamination, one hot pattern, a leaked template, pulls late transformer layers into recall mode. The model looks confident, but it is running a memorized circuit. Control and statistics. Cancer diagnostics control false positives across many tests. SCAR controls false discoveries across layers and prompts. We do not call a cancer because one pixel is bright. We call it when the ring, the uplift, and the trajectory kink all line up and pass FDR. A simple story to say over the clip. Biology shows how a tiny change can overactivate a pathway and lock a system into a bad state. LLM contamination does the same inside a network of layers. SCAR looks for that overactivation in the model's geometry. If we see coactivation in the late layers, a closed rim in the isosurface, uplift on the semantic earthquake map, and an eruption on the summary surface, we call a rupture. Otherwise, we call it clean. One-line takeaway. Oncogenic overexpression in cells leads to overactivation of a memorized latent pathway in LLMs. The video shows the biology. SCAR gives you the instruments to see the same failure mode inside a transformer.